Now, the final speaker is Addie Thompson, who is from Purdue University. She's going to try and now tie in some of the phenotyping technologies with genomics and look at how these technologies can be applied to try and address some of the challenges for food security. Her work has been focused on drought tolerance in maize and I think also sorghum to some extent. And she's going to talk now about addressing the challenges of global agriculture using advanced plant traits. So, Addie, over to you.
Thank you, Peter, and thank you, Morris, and everyone, and Tala, for the invitation to come and speak today. I'm very flattered. And thank you to all of you who stuck around for this late session. I know that you're probably getting a little bit tired. As Peter said, I'm going to try to give sort of a bird's eye view. We'll get into a little bit of details, but it's more of an overview, I think. And I could consider myself, I've been trained in plant breeding. I wouldn't call myself a breeder to any breeders, but in this context, that might be the closest approximation. Plant geneticist, you could say. So the first thing I wanted to say was that in a phenotyping project, especially in high-throughput phenomics, it's important to go through an order of operations, right? So the first thing to consider is, what are the issues? Are you studying drought? Are you studying disease resistance? Are you breeding for yield? What is the issue that you want to address? And there is a lot to be said for research that collects a lot of traits on a lot of different things sort of indiscriminately because investigative research is still important. But in the context of a breeding program, breeders are not going to be interested in 1,000 traits. They're interested in what they want to use to address their question. So what are the issues? The next thing, what biological questions can be asked to address those issues? The next thing that has been mentioned a couple times is, what germplasm are you investigating? This is an important question that needs to be considered. So one approach is like what James does, where you pick something that a lot of other people are studying, right? And that way you can leverage that material and you can leverage their data in analyzing your own data, which is fantastic. Another approach that was brought up was to bring in diversity, and that can include wild relatives. But the main thing breeders need to choose from is variation, right? You need variation to select from or you're not going to make any progress. And the other thing is that it also needs to be relevant germplasm to, again, the biological question that you're trying to ask. The next thing to consider, what traits are you collecting and when are you collecting them? That means that you need to consider your experimental design. You have to plan in advance. You have to have contingencies. There's always going to be something that happens. But if you don't plan what traits you're going to collect and when, you might miss the one that is the most important by trying to collect other things that were less important, as it turned out. And then we'll get into the more complex things, how to integrate the various data streams. I know everyone's probably had this problem. You have a lot of highly complex data from a lot of different sources. And then how to model complex traits, and finally, how to transfer this knowledge to solve those issues that we addressed at the beginning of this order of operations. So that's the rough, I don't want to say outline of my talk, because I'm not going to talk about all these things, obviously, in detail. But that's sort of my perspective on bird's eye view of high-throughput phenotyping. I'm going to start out actually by sort of introducing the Tuinstra Labs projects that we're working on, just to give you a sense of what, I guess, a global breeder would be working on, right? Just some perspective on a breeding program. So here is the Bench Tuinstra Lab at Purdue. This is where I am. We're in Indiana, in the US. And some of the things that we work on in Indiana, we look at stomatal density in maize. We look at sorghum and maize grain protein digestibility, sorghum kernel composition. We're breeding some popcorn dwarfing lines. And this next one's kind of interesting, sorghum during metabolism modification. So sorghum is a popular forage, which means that you can grow it and animals can eat it, and then that serves as their calorie intake. And then sorghum actually grows back, unlike maize, where if you cut it off, it's just gone. Sorghum will grow back, and it also tillers highly, some varieties anyway. But the problem is that if you cut off or eat vegetative stage sorghum, it actually releases cyanide gas, which can kill the cattle that are eating it. So that's clearly not a good option. So in the developing world, I mean, it's known that you can't let cattle feed on sorghum until it's a certain age or stage of development. But something that the lab is working on is creating sorghum varieties that do not do that, so that they do not make the precursor to this cyanide called durin, and that enables the sorghum to be healthy for cattle to eat at any stage. So that's important, obviously, and we're looking at ways that we can actually screen that more effectively. Another thing is dwarf stable sorghum lines. It turns out that dwarf sorghum, I mean, it's highly important for creating grain sorghum that doesn't fall over, but the dwarfing events that are used widely are actually not stable, and they revert back. And it causes actually a huge yield loss. You'll have to excuse me, I'm getting over a cold. Another project that I'll talk about a little bit is one that was funded by the Howard Buffett Foundation. This reminds me that I'm actually trying to put my acknowledgements throughout the talk. So this is one of those cases. I'm acknowledging the Howard Buffett Foundation for funding this project. But we've been looking at maize drought response in elite temperate and tropical test crosses under controlled irrigation. So we grew corn in the desert and either irrigated it under well-watered conditions or drought, and then compared the two. We're also looking at maize kernel composition, seed ear photogrammetry, maize leaf membrane lipid composition, maize carbon isotope nitrogen discrimination, all kinds of things on these lines. And then in collaboration with some breeding programs in Africa, we have funded by the Bill and Melinda Gates Foundation sorghum functional gene discovery platform. So that is basically creating mutants in sorghum and then looking at how those can be used to do functional genomics. And striga management technology, and then finally the sorghum trait development pipeline. So this is a sorghum millet project. And then in collaboration with USAID, CIMMYT, and Pioneer, there's a heat-tolerant maize for Asia program that works in collaboration with the people at Hyderabad, India, at the CJR center there. I think that's all. Yes. And then, the reason I'm probably here is that we also work on an automated sorghum phenotyping and trait development platform. This is in collaboration with the Purdue engineering group. So we have a number of various types of engineers on the project, as well as plant scientists and aviation technology folks, remote sensing, GIS. And then we're doing this in collaboration with IBM Research at the Watson Institute in New York. So they help us with some of the machine learning and that sort of analysis. And then also, we've brought in a crop modeling component with some folks at CSIRO, University of Queensland. So we're working with the APSIM crop model, which I'll talk about a little bit later. But that's our way of sort of integrating the environmental data as well as the other work that we're doing. It adds some relevance. So the people we're working with there, so I don't forget to mention, Scott Chapman, Graham Hammer, and Greg McLean. All right, so of that massive projects, the two I'm going to focus on today are this Drought Tolerant Maize project, as I mentioned before, and high-throughput phenotyping in a broad sense going on at Purdue. So where are we at Purdue? What sort of functionality do we have? And then specifically with this ARPA Energy Terra-funded program with IBM Research and University of Queensland, I'll just briefly mention some of the things that we're doing. So, as I've said, the first thing we do is identify the issue, right? According to the crop science societies in America, the grand challenge of agriculture, overarching grand challenge, is that we need to double the global food, feed, fiber, and fuel production on existing farmland in the 21st century under conditions of climate change. And we need to do that in ways that do all of these things. It's no small task. Obviously, that's why it's called the grand challenge. But I highlighted under conditions of climate change because climate change is tricky. And the ways that we have in our toolbox to be able to adapt to climate change as plant breeders or as plant growers would be, first of all, you could change what species you're growing, right? So if you're growing, say, maize as a forage, you could switch to sorghum because it requires less water. And it can be a useful forage crop, especially if it doesn't produce hydrogen cyanide that kills your cattle. The second thing you could do is to modify your management. So change planting dates, tillage, double crop, leverage that G by E by M whenever you can. But the third thing, and this is what I'll actually talk about a little bit more today, because this is relevant to the work that I do, is developing varieties of target crops to improve adaptation to climate variability. So like I mentioned, heat-tolerant maize, but also drought tolerance in maize, and then developing better varieties of sorghum. So why are we studying maize? This probably doesn't need to be said. Maize is important, drought is important, but the part that I want to stress here is that drought is actually very difficult to study from a biological standpoint because, first of all, you have to define drought, which doesn't sound hard, but it really is, because drought can occur at different stages of the plant, and that has a different response. Drought can be at different levels, extreme drought versus mild drought. Drought can be in combination with other things like heat. And any of those sorts of combinations decrease the heritability even further from what it's already reduced under drought conditions. And then finally, the logistics. You can't tell the weather what to do, and so there's no way to make sure that you actually have a drought trial other than to grow it someplace like in the desert. But if you're hoping that your locations may or may not have drought, you could get lucky or you could not. The material that we chose to use for this project is called the Drought Tolerant Maize for Africa lines, and I wanted to take a moment to highlight this project. So I'll refer to this as DTMA, but this was a project that was started a number of years ago. It finished up in 2015, and the goal was, it was a collaboration between CIMMYT, IITA, and a number of large institutions and governmental organizations. But the goal was to breed new varieties of maize that would be useful for Africa, while also doing all of these things, right? Fostering seed markets, engaging government officials, providing training, increasing food security, providing support, all of these things. So it was meant to be a cohesive project, and these lines are actually grown fairly widely. But the exciting thing is then that we can use some of this material, leverage that for investigating for usefulness for our own purposes, right? So this is what we call elite tropical material. It was developed in Mexico for use in Africa. And so a lot of this stuff, if you grew it in the US, wouldn't necessarily probably flower in the right time because it's used to having really long days and really hot days. So that makes it an interesting challenge that I'll get to in a moment. The other material that we have, so this is the elite tropical lines. It's out of public breeding programs. The other material that we have access to is what we call the XPVPs, and that is expired plant variety protection. In the US, we have a plant variety protection program, which means that it's kind of like a patent, but not really. But basically, the variety is protected for some number of years, and then it goes off of that. And when it goes off of that, it's free for use for anybody. And so this would be material that had been used in places like Pioneer, Monsanto, Syngenta, any of those multinational corporations that had produced and released these lines that are now available for use. So what we did in developing our germplasm to ask these questions was we took these two sets of material and we crossed them to two testers. Now maize, for those of you that don't know, exhibits strong heterosis. So when we grow maize in the field, we usually grow hybrids. As a farmer, you would grow a hybrid because it has hugely increased yield. But the thing is, there's different pools of germplasm. So there's different heterotic groups. And if you cross two like things, you won't get as much heterosis as if you cross two different things. So what we did here is we took two testers that were very different and then crossed all the material to both of those. And then obviously we grew it in drought and well-watered conditions. So what that gives us is diverse elite material that we're starting with from both public, tropical, and private temperate material, evaluated as hybrids, so a relevant context, under relevant field conditions. So that's pretty exciting. And the goals here are, first of all, just to characterize the lines and their response to drought, again, in relevant genetic and agronomic backgrounds, and then to identify unique regions of the genome that may be beneficial for improving maize in either context, right? So we could find some temperate material that might be beneficial to improving the tropical germplasm, or we might find tropical material that's beneficial to improving the temperate germplasm. Either way, it's an interesting question. And this is my lovely cartoon of what I just described. So in, say, the temperate material, we might have some positive allele on this chromosome here, and in the other material, we might have something different. And by crossing the two together, we might be able to get a combination that's more beneficial than if we had crossed within either group by itself. very, very basic breeding diagrams. And so how did we evaluate this material? We took a number of sort of basic phenotypes, things that breeders would normally take, flowering time and plant height and ear height. We took a chlorophyll content meter reading for a stay green. So that's basically after some of the plants start to senesce, how long do some of them stay green? And then a few yield components. We also took some more advanced plant traits. We looked at leaves to model traits, leaf membrane lipid composition, carbon and nitrogen isotope ratios. These things are getting at the drought response, and water use efficiency, which is why we took those sorts of traits. The idea here, branching off of my previous breeding cartoon, is that we're considering a new breeding paradigm here, right? So breeders tend to cross the best material to the best material. And whether the best is defined by the phenotype itself or by a prediction out of a genomic prediction model, either way you're looking at the highest number that you're trying to cross to the highest number, right? That can lead to, though, you're limited by your inputs, right? You can only get the best of what you had to start with. And so if you have these two, let's say loci, that are fixed as a red, meaning negative version of that allele, right? You can only ever get as good as what you started with. Instead, if you took these two lines, where they have actually maybe a little bit worse scores to begin with, and cross those, you have the potential to get something much better in the end. Now, this is pretty practically impossible from a breeding point of view if you're thinking about buying lottery tickets, as someone said earlier, right? You'd have to buy so many lottery tickets that this is pretty much not viable. Well, I'll get to that in a moment. But for now, think about the idea that you could be combining things that might not look as great, but if they're involved in different loci that are controlling the trait of interest, if they're involved in different physiological processes that are leading to the same positive trait in the end, you could potentially get better improvements by combining. So, how is it that we could actually visualize a chromosome in that fashion? This is work that I did with Rex Bernardo a few years ago, and the idea is that, you know, as a breeder, you have an idea of what germplasm looks like what as far as the numbers, but you don't have a good concept of what things look like as far as what's going on in the genome with marker effects. So I feel like a lot of people would have a good sense of, you know, this region of the genome controls this trait, and these genes have this effect, or these different types of material have these types of traits. But the connection there is missing a bit. And I'll try to explain what I mean using a little bit of statistics, simplified. So imagine a SNP matrix, all right? You've got a matrix of basically ones and negative ones for either SNP, zeros for the heterozygotes, and you have those as, you know, numeric genotypes. Then you have done your genomic prediction, and your model comes up with effects for each of these SNPs. So you estimate an effect for each SNP, and you say, this SNP has this effect on this trait. So maybe this SNP has 0.03 days that it adds to the flowering time, for example. Now, if you multiply those together, you get a numeric matrix of SNP effects. So you're basically just positive A or negative A, or 0. Now, if you take a rolling average, or you sum across a region, or even a chromosome, what you end up with is something that looks like this. And you can say, okay, for inbred line A, I have a really poor version, let's say, of chromosome 1, but, or in individual B, let's say, we have a really poor version of chromosome 10, but the rest of it looks pretty good. But you can start to see how if you were to cross good by good, you know, maybe it would be better, maybe it wouldn't, but there could be, the highest complementary would be if you crossed it with C, which overall looks pretty bad, but it's got that really high value for chromosome 10. So this is just a way of shifting the thinking of the overall value versus adding up pieces. And as I said, this may seem like it's not very viable, but actually Rex Bernardo, this last year, came out with a paper where he was discussing breeding for quantitative traits using CRISPR-Cas9. So the question is, if you could direct just one recombination event, or if you could direct 2 recombination events, what kind of an increase would you see in yield versus if you were taking random crosses of the population, right? Or a random cross, but a directed cross, but whatever random individuals are created by that cross, right? So the idea that we could direct 1 or 2 recombination events in a cycle and use doubled haploids, that becomes huge. And this becomes not a pie-in-the-sky idea, but maybe something that's more viable. So what does this look like? If we apply this to the population I just talked about, this is now individuals across the top, hierarchically clustered, and it's chromosomes along the side. I know this is really overwhelming, actually, for a figure, but bear with me. So this is the marker chromosome effects by chromosome. And what I did is I allowed them to cluster on their own by effect, right? And what I did was I used anthesis under drought and allowed these things to cluster. And what happened was that my temperate and tropical lines clustered separately, except for that batch of stuff in the middle. That batch of stuff in the middle is material that is from the temperate XPVP germplasm, but it's behaving more like the tropical material. So that was very interesting. The other interesting thing is I only see this when I look at this under drought conditions. This does not happen under well-watered conditions. The 2 material sets don't separate like this. So it's a response to drought that's differential, and that becomes very interesting. So having ways to just visualize what's happening in the germplasm is incredibly useful and powerful. The second thing I'm going to talk about in this population with the data is a systems biology approach. Caveat, I'm not a systems biologist, but I played around with this, so it's hopefully interesting. The questions here, though, are what are the relationships among the genes that are involved in the control of these traits? And then what regulatory networks contribute to, say, drought tolerance, and what functions do they perform? The way I did this is using a network analysis, which means in any network analysis, you're taking a large data set, you're relating those data points in some way to find a pattern, construct networks, and then you extract some functionality and meaning from that. I think Kevin called this a spider web. I like to lovingly call it a hairball. We start with our hairball here, we construct a network, and then try and extract some sort of meaning. And the way that I did this was to use what's called a weighted interaction SNP hub network. So it's relating the SNPs to each other in a network. And what we find, if we do a topological overlap matrix and tree cutting, so this is basically just clustering the SNPs, and then you're identifying modules out of that clustering to say that these SNPs are related to each other and might share some sort of functionality. And so this is SNP clustering for days to anthesis under drought for the tropical lines. I did that because under drought and in the tropical material, that's where the heritability is the lowest. So I tried to make it as difficult on myself as possible. And what happened was I identified a number of these colored modules here. So this is significant chunks of SNPs that are related to each other. And what you can then do is for each of those chunks of SNPs, you can create what's called an eigensnp. So it's like a representative version or score for that cluster. And then those eigensnps you can relate back to the original phenotype. And what happens is that we see really, really high correlations. So this blue module, for example, the correlation is 0.69 with incredibly low p-value for relationship to anthesis under drought. All these modules are highly significant, but that's pretty interesting. So what are the genes in that module? We can look in and look at what genes are there and what their function is. One way to look at this is to look at what we call go-annotation terms. So each gene has a set of defined terms that it has associated with it, and we see things that are involved in drought stress and water stress and that sort of thing. So that becomes very interesting. I included also there was someone that was working on medical subject heading terms. This is an alternative to go analysis, but it comes up with sort of the same picture in that it's mostly drought stress related things. So I think that's what I have for genomics for today. Eventually, we can relate these SNP hubs to gene co-expression networks. We can seed the modules with known genes for specific traits. All kinds of other things we can do that I'm excited about, but I'm going to put that on pause for a minute and return back to this concept of the grand challenge of agriculture. I feel like the cutting edge technology in genomics is kind of keeping pace. You know, we're doing fairly well, but the problem is, as we all have been working with, phenomics, maybe not as much. So traditional crop phenotyping is slow, laborious, limited, sometimes dangerous. We talked about sometimes variable accuracy. This isn't to say that people who are doing traditional crop phenotyping are doing anything wrong. It's just that it's hard, and we recognize that because we do it. In fact, I think that's me right there. So this is where we get into the high-throughput phenotyping that I'm going to discuss for a little bit as part of the Purdue Smarter Agriculture, and then this Terra project I'll discuss a little bit. We started scaling up originally by renting a shed that we dropped down in the middle of our field, and we built some capabilities into it so that we could take controlled images of some of our plant parts. And the reason we're doing this will become apparent a little bit later, but the reason we're taking pictures of these parts like this, in that center, yep, is so that we can get a sense of the leaf area and the leaf area distribution, and also panicle size there. So here's the panicle of the sorghum plant, and then this is all the leaves of the plant laid out in order. And then we also weigh all of those parts, and we dry them, and we weigh them again. We take a number of traits on the plant itself. The other thing that we've been doing is using in-data field collection on electronic devices rather than clipboards and paper and pencils. This met with some resistance, but we have persevered, and I think as new versions of the app come out, this is actually Jesse Poland's Fieldbook app, so he's out of K-State. We found it fairly useful, and that has been helpful for at least putting all of our traits into one place, into one database. But the next generation is this idea of automated data acquisition and high-throughput phenotyping. We have a number of different vehicles that we're using, and this is it's a modified high-boy sprayer. We call it the Pheno Rover, so that has a number of sensors mounted on a custom boom, and that can drive through the field. However, things that drive through the field can only drive through the field if it's not too wet, and the plants aren't too tall, and there's not too much tillering, and, right? So ground-based phenotyping is fantastic, and it gets really high resolution, high quality images, but you're actually more limited as to when you can do it. We found that flights, so we have fixed wing as well as rotocopter type drones that we use. We can offload the data. These have this Telemaster here has a metal box in the bottom that sort of drops in and out so you can change your sensor payload. But those have been more reliable for as far as when we can actually collect data. I do have a picture of a biomass harvester here in the corner, not because it's fantastically glamorous, just because that's my reminder to say that sometimes phenotyping can be dangerous. This is a bunch of spinning blades here that chop off the material and suck it in. So you have to be careful to not be in front of it. All right, so what are we doing at Purdue? Purdue Smarter Agriculture. Basically, they're trying to turn the field at Purdue, the entire research field, into a precision agriculture zone, which sounds a little bit ambitious. But the model is, I think it was James that was talking about how plant breeders are subsuming more and more types of research. And one of those was bioinformatics. At Purdue, we found this to be sort of true. So what happened actually was they took some people out of the core facility for bioinformatics and moved them into a core facility for phenomics. So now we've got a core facility for phenomics where if you opt into this, your field will be flown once a week. They collect data for you, they do some pre-processing, they turn it into some numbers, and they hand it back to you. You get data once a week throughout the summer, and for some small fee. This year, they're doing it on a 30-acre area for free. And the idea there is that any researcher that is not sure whether phenomics would be of use to them or not can have some sort of buy-in to get some sort of data so that they can start looking at it and ask questions about, well, how would this improve my selection? How could I model the traits I'm interested in better? And I think that's really important. I will say also that if you're interested in convincing plant breeders to uptake technology, it's not so much about telling them that it's more accurate. High numbers are good, more accurate for their current traits. It's a good argument, but it can also come across as condescending if their measurements aren't good enough. But the selling point, I think, is that you can collect traits that are not being taken or that they might not know would be useful. And that's a harder thing to convince, but I think that's the more important route to take. The other thing is that there are a lot of traits that are really complex or are very hard to actually measure. And that type of trait is specific to the crop. So you have to figure out for that crop that you're working with, what are those traits you can use to convince a breeder, right? A lot of times it's something that has low heritability, it's hard to screen for, it's expensive to screen for, so think about something like disease. If you have the investment in a field trial to actually impose the diseases you're looking at, you want to get the best data that you can and any types of data that you can. One of the most convincing talks that I saw actually was someone that worked with potato, and he was able to go in with hyperspectral imaging and show diseases that were occurring in the potato plant underground. So if a breeder were to go out and look at the material to screen it, you cannot tell a difference. The leaves look exactly the same, but the potato itself has a fungal pathogen, and the leaves have a different hyperspectral response before you actually see it visually. And so that becomes something that's really convincing, especially for someone that may need to apply a fungicide to prevent the problem, to be able to see that in advance. So that's my 2 cents on that topic. So how are we getting data? We are using a number of platforms, as I mentioned. The main sensors we're using are LiDAR, which is lasers that spin and bounce off of the material and then give you a point cloud approximation of your geometries. Hyperspectral imaging, and then RGB and high resolution. We'll be adding thermal eventually. All right, this is a very complicated slide, but I wanted to take the time to go through this to give you a perspective on the plant science side. So the sensor technologies I mentioned on the left, LiDAR, reconstructed RGB, hyperspectral imaging, thermal imaging, it's tempting to try and use whatever sensors you have to try and predict whatever's going on over here, right? So how do I use this and then get at total biomass and yield? The problem is there's a lot that's going on in between, right? There are a lot of interactions between genes, with the environment, any number of things that are going on in between there. So what can make more sense that we like to try and do is that we use these sensor technologies to either measure or model some of these traits in the middle. So for example, you can take something like reconstructed RGB and hyperspectral imaging in combination to measure something like canopy cover, which does happen to be directly related to total biomass, but there are other things that, that gets taken into account. I will mention that we do have here listed biochemical composition. That's come up a few times, and we are working on right now looking at lab-based biochemical composition from stalks and leaf material, as well as NIR, and relating that to what we see from hyperspectral of both leaf clamps and aerial data. I don't have any results on that yet, but what I can say that's interesting is someone mentioned lodging resistance earlier as far as in relation to composition. Yes, that's true. We see some colocalization of QTL that have to deal with biochemical composition like lignins and sugars in the stem with lodging. So something that's interesting. Let's see. So what is involved in collecting these data sets? As you probably all know, lots and lots of processing and calibrations and correction. So this is just an image of one of the drones flying over these wooden targets out in the field. These are just boards, basically, pieces of plywood on a plank. And so the idea is that you're flying over it multiple times and trying to get these little dots here to line up perfectly. And that's especially important when it comes to something like LiDAR. So a LiDAR point cloud that you get if you drive back and forth over it, if you're not calibrated properly, it can look like it's ghosted as multiple things. If you calibrate it properly, it will become one piece of wood, right? Imagine that being sorghum stalks or plant stalks instead of a piece of wood, and you see why that's really important. This has come up before, but using some targets out in the fields, flying, we have X and Y accuracy of about 1 centimeter, and the Z is 2 centimeter. And the trick then is this co-registration of the hyperspectral images. So our team has developed a stepwise orthorectification procedure, which is published here. And that allows us to have basically pixel level hyperspectral images that are perfectly aligned with our RGB. So we're currently developing this platform in sorghum because that's where that was funded to be. But the plans are then to apply those approaches to the DTMA and XPVP maize that I mentioned earlier. And as was also mentioned before, there are lots of ground validation studies that have to go into creating big data sets like this because we need something to model against. However, we are careful not to call it ground truth because, as we have found, there's several reasons that it's not necessarily the truth. One could be that your ground measurements are not accurate, which does happen. But another could be that you're actually measuring different things. So someone was talking earlier about when they're looking at roots, if they go out and they wash off the roots, then they've added water to the mass of the roots. And so that's no longer the actual appropriate measurement. So you have to vacuum it off and then, so you have to consider these things and remember that, you have to compare apples to apples, not apples to oranges. But there is a lot to be said for data QC and quality control, and the use of really good statistics and good experimental design so that you know what data to keep and what to throw out. But I will say that, add the caveat that high-throughput phenotyping is about more than just replicating what breeders can already do in the ground. It's about adding to that and adding traits that may not be observed otherwise as well. One example that I like to give, and this has come up a little bit already, but it's really dramatic when it comes to sorghum. So as you are flying over the field, you get overlap in your images, and that allows you to calculate not only the X and Y position of each pixel, but also the Z, so you can reconstruct these canopy level images, as was mentioned before. And what that ends up looking like is something like this. And here we have actually, if you're familiar with sorghum at all, this is the Bioenergy Association panel. So this is about 370, 380 lines or so. And this is material that was bred to be high sugar content, high moisture content, really, really tall, good for bioenergy, but not great for agronomics. So it grows to be like 5 meters tall. It tillers like crazy, which means it grows lots of stalks. So any one plant that you plant grows to be, a big mass of stems. And then this material here is what we call our calibration panel. What we've done is we plotted really large plots here. You can see they're quite large. They're 12 rows actually, because those are the material we go into when we take destructive samples for calibrating our biomass models and that sort of thing. But that material is 18 commercial hybrids. That's hybrids that are grown today that represent a wide variety of different types of sorghum, which is why they're all wildly different heights. The shorter ones are the grain sorghums, the taller ones are like the forages and the bioenergy type material. So this was taken on, I should mention, this is actually corn, so that's why it looks very different. This was taken on August 9th of last year. What happened a few days later is we had a big rain event and a big windstorm, strong winds that came through. This was how it looked later. So as you can see, a lot of it has now fallen down. Yay, lodging. This was depressing because it was basically a total loss as far as what we could get on the ground. We couldn't get any ground truth data anymore, right? You can't get ground calibration data. We tried really hard. We had somebody sprain their ankle, we had somebody twist their knee. You can't physically walk through a field of lodged sorghum. You can't get to all the plots. That's something people don't think about, is that high throughput phenotyping type approaches where you're having remote sensing, one of the advantages is you can get places you couldn't get otherwise. And if you've ever tried to walk through a fully grown field of sorghum, you can appreciate this. Walking through it even before it lodged was difficult because it's like a jungle, but walking through it afterwards is pretty much impossible. So I'm pointing out here that, as you can see, there's a few plots where obviously they lodged, and there's a few that were still standing afterwards. And so looking at this from the optimist point of view, this gives us a good opportunity to look at what genotypes are more or less likely to lodge, and we can look at lodging resistance, which is great. So ongoing work with this project, we're using predictive models. We're developing predictive models using the remote license features. We're also interested in looking at interplot variability features. So one of the things that people tend to do is they tend to get one value per plot. We're actually interested in looking at how those values change within and across a plot, and I'd be happy to talk more about that at some point if anyone's interested. We're using machine learning approaches to link genotype to phenotype, using high-throughput models, high-throughput phenotyping to parameterize crop growth models on the individual genotype level. This one's a little bit exciting, and I'll go into that a little bit more. So this comes back to the question of how do we incorporate environmental and developmental differences? Because what is happening to any one genotype at a given time point isn't necessarily the same thing that's happening to another given genotype at that time point, because it's a different developmental time point for any given variety. So the question becomes, what is happening to the plant on a physiological level throughout the course of the season? This allows us to also ask questions about stress. What is the impact of the stress and why? And how does this plant growth, development, and response vary by genotype? Someone mentioned earlier today, Peter, I think mentioned using flowering time as a fixed effect in your models. That is the very beginning of this. I would argue that gets to the bare minimum that you should be doing. But on a broader scale, there's a huge impact of what's happening on any given day with where the plant is at. As the crop modelers like to say, this can give you the right answer for the right reason. Right? So you might be able to find what has the highest yield in the end, but why does it have the highest yield? What happened throughout the season to cause that? And are there other things that got to the same result via a different mechanism? And the idea, in the end, is that we want to be able to transfer our predictive models to other regions and climates. So it's great to predict what's going on in Indiana, but it'd be really great if we could use that model elsewhere. And the goal then is to combine information from crop models, genomic prediction, and high-throughput phenotyping all together. So this is a sample of what we're doing. We're using a crop growth model called APSIM. Now, if you're not familiar with crop growth models, the inputs here are that you're taking weather data, so rain, sunshine, temperatures on a daily basis, and this is part of the model then that gets fed into the process as far as how the plant is growing and changing on a daily basis. So the blue lines that you see are the output on a daily basis from these models. And I have it set here to show the accumulated biomass, the canopy cover, what we call the leaf area index. That's a measure of how much leaf area you have in an area, and then grain yield. And the bars that I'm showing are ground truth, ground calibration data that matches up very well with the model, what the model is predicting that we should have. So the question is, how did we parameterize this model? What we did is we took those leaf images that I showed you earlier, so the board that has the leaves laid out on it, and we looked at the size and shape of those leaves. And it turns out you can model those as a power function across the plant. And we can use the different parameters that we've looked at in the field, so how fast do those leaves appear, what the size and shape are, and use that to parameterize the crop model on the level of each individual genotype. So again, if you're not familiar with crop models, what's normally done is it's parameterized or it's developed for a crop, right? So you have the corn model, and in our case, or the sorghum model. But here what we're doing is we're actually changing the parameters for each individual genotype and then seeing how that performs and how we can actually predict what's going on from a breeder's perspective. So the future then is we're going to parameterize these crop models for thousands of varieties, eventually with remotely sensed features. So we're using our sensor data as opposed to our ground truth data, and then combining that with genomic prediction to inform our crosses. So what does that look like? Actually, I'll go back one. What that looks like then is that you would be potentially parameterizing a model using remotely sensed features that are predicting more heritable inputs. So let's say you used remote sensing to predict something like canopy cover, or you predict something like the number of leaves that it has in the end. And those are things that can be modeled pretty readily and things that are also highly heritable and not as sensitive to the environment. So if you take specific basic inputs and then use a crop growth model actually to combine that information with the soil parameters you're using, the weather that you're looking at, and actually get a more informed prediction in the end. The fun part then is combining this with genomic prediction, right? So you can train your models on some genotypes, predict genotypes that you haven't observed yet, and then predict how those are going to perform in environments that you haven't observed yet. And that's the real gold bar we're aiming towards. So, high throughput traits, the idea is to collect information like plant location, spacing, leaf number, canopy cover. You can read the rest of these. I will highlight also latent features, so things that we don't even know what they mean, but we can pull them back out later from images and ask what it is that we're actually looking at. That's exciting. And I also want to quickly highlight plant location and spacing. This sounds really basic, but it's something that planter companies are highly interested in to know how well they're doing. But it's also hugely important because plant density and spacing impacts everything else you're looking at. So if you're trying to model something that's occurring in your plant and you have a different spacing than what is used, say, in a breeding context, your models are not going to be at all predictive. And we know this because in our calibration panels, we had two different densities. One was 52,000 plants per hectare, and one was 223,000, which is actually the commercial density for sorghum. So that equates to about this far apart versus this far apart. And sorghum is interesting because when you plant it less far apart, it actually tillers more, so it produces more stems, which means that by about midway through the season, we couldn't physically walk through the low density, and we still could get through the high density. So it actually creates more biomass in some cases. But that made it so that our models were not at all predictive from one to the other. So that's something you have to be careful about. So using high throughput traits, using our statistical approaches, and including predictive modeling, crop modeling, genomic prediction, using these to create enhanced outcomes of innovations. I won't read the slide to you, but the end results are that we're hoping to get a better understanding of plant genetics and development, as well as creating optimized crop varieties and production systems for food on a global scale. So, how do we do this? We collect phenotypes in an informed and targeted fashion, train genomic prediction models on our key traits that I mentioned, like things that are not highly responsive to the environment. You can parameterize a crop growth model on a per genotype basis using those predicted parameters. That can also include predicted and simulated genotypes, as I mentioned, so it could be genotypes that you could create in your breeding program, hypothetically. Use your model to integrate the soil, climate, and management data with the component traits to predict the compound traits, like yield. And that allows you to transfer predictive abilities to other locations, soils, and climates around the world and into the future for climate change. And this is a slide that I blatantly stole from Pioneer, from a presentation that was shown online. But this is work done with Charlie Messina and Mark Cooper, and they're highlighting the idea that we know that yield is controlled. Yield is a function of genotype, an environment, and a management. This is what the GEM stands for, genotype, environment, and management. So if yield is a function of those things, we can use, say, a crop growth model as the function that incorporates those things. I will say there are skeptics about crop growth models because they can be kind of complicated. The beauty of this, though, is that once we're getting into the high throughput traits, the high throughput traits actually take into account some of the complexities of the crop model. So the goal, I think, eventually is to simplify that down, and so that we're not using the crop model directly, but we're using kind of a simplified model where we say, all right, if we can just look at, say, height, canopy cover, and canopy temperature. I've heard Jesse Poland give talks before where a model that incorporates just those three things actually accounts for a huge amount of the variation in yield in wheat. So that's kind of the end goal, is to simplify it back down and be able to predict these things on a large scale using a simplified model. An example here, again from Pioneer, that they used that I found incredibly fascinating was they took one of their Aquamax varieties and then a bunch of their pre-commercial products. And they said, all right, in 2012, we had a horrible drought. In 2012, how would these varieties all have performed relative to each other? So they can plot this out across, their growing region and say, are any of the varieties that we're currently producing in our pipeline going to outcompete Aquamax? Can we do better than what we have already? Or is that as good as we're going to get? So this is what is being used. And here it's being shown for pre-commercial predictions, but you could just as easily use this for simulated genotypes. So if you use genotypes that don't exist, but you have genomic prediction for them, you use genomic prediction to predict your input traits, your parameters, and then use some sort of crop growth model to integrate that to be able to predict. The long-term goals and applications on the bottom line are that these new technologies and approaches will not only enable gene discovery, but also inform future research and breeding efforts to optimize crop varieties and production systems. The idea being that the innovations that we're making in Purdue University and in the US, and these models that we're building here, will be transferable to places like Africa and Southeast Asia. And you'll notice this map looks really similar to the one that I showed earlier. The one I showed earlier was actually water stress. This one is nutritional deficiencies, but they're very similar. So it's the same regions that are having the problems. And I'm going to end here with my view of sort of what all is involved in addressing these issues. As everyone has said, you've seen this equation now a lot of times. The breeder's equation, we need people that can do remote sensing, high-throughput phenotyping. We need people that can do lab work, field work, genomics and genetics, high-throughput computing, everything. We also need coffee. So thank you. I'm happy to take any questions if there's time.